Hi friends, Namaste. I am Varsha Gaitonde and welcome to my channel. This video is about molecular basis of heterosis. Molecular basis is explained at two models which are used to explain the heterosis. One model which consider that in hybrids having two different kinds of allele, an allelic expression in additive manner occurs. And the second model that states that the combination of different alleles cause gene expression changes in hybrid that deviate, which is related to the mid-parent. This concept of dividing the model with the help of molecular level is given by Birchler and co-workers in the year 2003. There are different mechanisms to support the molecular basis, like the first mechanism is intermediate amount of a single gene product. This is explained in case of neurospora with PAB genes. PAB is responsible for the production, or PAB means p-aminobenzoic acid. When mutant form of PAB is there, it gives no effect, means there is a reduced effect, and when wild type is there, it is also having some different effect. But when the mutant and wild forms are brought together, that gives faster growth rate in case of neurospora. Second mechanism is separate gene product. Two alleles of a gene produce different gene products. The heterozygous are more adapted to changes to environments than the two homozygotes. For separate gene products, the example or best example is sickle cell anemia, which is caused in homozygous recessive state. That means homozygote recessive will die. But the heterozygote condition in many of the African countries, it is resistant to malaria. The third mechanism is combined gene product. Many proteins are homodimers or homopolymers. The hybrids are made up of two or more polypeptide protein molecules with different properties. The combination may form a new gene product. Example, two alleles of a gene produce octanyl dehydrogenase 1 enzyme responsible for heat sensitivity in homozygous form. But in heterozygous form, it is less sensitive. Heterodimers have less sensitivity in case of maize. Fourth one is effect in two different tissues. ADH is involved in production of superior biochemical product due to balanced rate of transcription due to the presence of alternate forms of two tissues. Example is ADF has high activity in seedling tissue and ADHS is having high activity in pollen. Here you can see the seedling and pollen are two different tissues. When ADHF and ADHS are brought together, that gives higher growth and vigor during the development. The fifth mechanism is expression in greater amount. Heterotic hybrids show 30 to 60% better gene combinations due to reduced methylation pattern than compatible mRNA level, better post-regulatory expression if it is over-dominant, then housekeeping genes in case of dominance condition, and better activity of indels. Indels means insertions and deletions. In case of maize, number of genes expressed in hybrids are usually 30 to 60% higher than those of their parents, and heterotic hybrids express as a much higher number of genes than do non-heterotic hybrids. This enhanced gene expression may be due to transcription factors encoded by QTLs showing overdominance. So, in the molecular pattern, the expression of genes in different amounts is mainly the thing, and here it is affected by methylation pattern or mRNA level, better regulatory expression, housekeeping genes, and better activity of indels. That means the molecular mechanism is supported by the biochemical factors. So far, whatever the 5 mechanisms I have discussed, those are related to the biochemical factors, but in relation to that, when the expression comes, there are different things. Here we will discuss one by one. The levels of expression of the gene. Comprehensive analysis of gene expression reveals that most of the genes that are expressed at different levels in the parents show intermediate level of expression in hybrid, suggesting the additive action. In the previous slides, I was discussing about the intermediate level of expression of the certain substances, and that is reported as additive action, but in some other cases that is non-additive also, that is dominant and over-dominant genes in hybrids, suggesting transacting factors like transcription factors play an important role in heterosis. The proportion of genes exhibiting non-additive expression is mostly small, but in some studies it is relatively higher. This variation depends on species, parental genotypes, tissue analysis, and experimental design used. Hybrids show non-additive expression patterns for proteins as well, and the proportions of such proteins and the type of non-additive effects observed vary substantially among different studies. First one is additive expression. Additive gene was prevalent in some studies like maize. Non-additive expression was different in reciprocal triploid hybrids but same in reciprocal diploids. It may be indication that dosage of different genomes alter the nature of non-additive expression, suggesting the role of regulatory effect. QTL mapping is one approach which gives a definite pattern of expression of particular genes. That means when we do the QTL mapping, we can find out the particular position of a gene and as well as its expression pattern. When transcript level is measured in a mapping population and treated as a quantitative trait like any other trait of QTL analysis, then it is described as eQTL. The expression QTL controls the expression level of transcripts. Remember, the expression QTL controls the expression level of transcripts. When QTL affecting gene transcription maps close to the affected gene, it can be classified as cis-acting, and when QTL that maps further away on the same chromosome or to another chromosome, that is classified as transacting. Means the cis-acting and transacting nature of particular QTLs decide the expression or the pattern of expression. नेक्स्ट वन इज़ एपिजेनेटिक वेरिएशन. एपिजेनेटिक वेरिएशन के बारे में बताऊं तो इसके लिए एक सेपरेट वीडियो है एपिजेनेटिक्स के ऊपर, तो आप वो देख सकते हैं. लेकिन अगर मोटे मोटे तरीके में एपिजेनेटिक्स के बारे में बताना है तो ये क्या होता है, दिस इज़ ए चेंज एट सरफेस लेवल विदाउट एनी अल्ट्रेशन इन द डीएनए लेवल. मतलब एक्सप्रेशन में चेंज होता है लेकिन डीएनए या बेसिक लेवल में कोई चेंजेस नहीं होते हैं. एग्जाम्पल क्या है, डीएनए मिथाइलेशन और हिस्टोन मॉडिफिकेशन डिफरेंसेस इन अराबिडोप्सिस, मेज, राइस जीनोटाइप्स that can exhibit stable inheritance. There is also evidence for natural variation in small RNA abundance among individuals of the same species. मेज़ के ऊपर स्टडी और अदर क्रॉप्स के ऊपर स्टडी ये बताता है कि जीनोमिक डीएनएस ऑफ इनब्रेड्स आर मोर मिथाइलेटेड देन दोस ऑफ हाइब्रिड्स एंड दैट डीएनएस ऑफ हेटेरोटिक हाइब्रिड्स आर लेस मिथाइलेटेड देन दोस ऑफ लेस हेटेरोटिक एंड नॉन हेटेरोटिक हाइब्रिड्स. ये मैंने बताया है. इन एडिशन डिफरेंट जीनोमिक रीजन्स में बी हाइपर मिथाइलेटेड इन डिफरेंट इनब्रेड्स. It is believed that the plants that level of DNA methylation is negatively associated with gene expression. मतलब ये पैरेलल में नहीं चलता है, ऑपोजिट टर्म्स में चलता है. कौन सा डीएनए मिथाइलेशन एंड जीन एक्सप्रेशन. मतलब डीएनए मिथाइलेशन का रेट ज़्यादा है तो जीन एक्सप्रेशन कम हो जाएगा, और जीन एक्सप्रेशन ज़्यादा है तो मिथाइलेशन पैटर्न कम हो जाएगा, और इसी को हम हाइब्रिड्स में देख सकते हैं. Therefore, reduced level of DNA methylation in hybrids may lead to increased gene expression and consequently heterosis. Epigenetics is another field which leads to heterosis, or it's another cause for heterosis. As you know, epigenetics refers to heritable through mitosis or meiosis alteration in gene expression that are independent of DNA sequence. Different epigenetically regulated forms of a gene are known as epialleles. So, the types of epigenetic expression on heterosis are DNA methylation, histone modification, and RNA interference. Histone modification leads to chromatin remodeling, that means changes at a histone level, and RNA interference means with the help of siRNA, miRNA and shRNA alteration at the level of gene expression. Epigenetic information system could generate epigenetic variation or epialleles that had never been considered as the cause of phenotypic variation. Local chromatin status mediated through epigenetic modification can potentially affect gene expression in cis or trans regulatory model. Next is RNA interference. RNA interference is a conserved mechanism for regulating gene expression. This is the major process which occurs through double-stranded RNA. Here, three things comes in RNA interference. One is miRNA, then siRNA, followed by shRNA. All these are having their own activities which are negative regulators of the target transcripts accumulation. Mediate post-transcriptional gene silencing, RNA-directed DNA methylation and chromatin remodeling are the basic activities of these RNA interference mechanisms. Here one thing you should note down, miRNA and siRNA are differentially expressed between hybrids and its parental inbred lines. If siRNA and miRNAs do not match genes from one inbred to the other inbred, the resulting hybrid could exhibit novel pattern of gene expression. Long non-protein coding RNAs, that is NCP RNAs, are identified as precursors of mi and siRNA. That means their presence in different amount or their expression in different rate gives better expression pattern in case of some of the hybrids or better heterotic expression. Here in this picture you may see the potentiality of heterosis. As in parent A, the transcription factor is inactive. It is unable to carry out the transcription process because of some things like it is unable to bind the promoter region. The same condition is observed in parent B, but when in F1 hybrid is produced, you may see that transcription factor is able to bind to promoter region, and finally there is a transcription process. This is how the DNA methylation process works on the process of heterosis. Conversion of cytosine to 5-methyl cytosine could generate epigenetic variation, epiallele and creation of hybrid vigor. DNA methylation does not change the DNA sequence and its function, but does change its expression level, referred as epigenetic change. Association with gene silencing and genes with abundant 5-methyl cytosine in the promoter region are usually transcriptionally silent, function to suppress as regulatory factors. This is the reports of different scientists as given. I have compiled and given here. Altered regulatory interactions could play a role in the increased vigor. Basically, heterosis is the result of different allele being present at loci that contribute to the regulatory hierarchies that control quantitative traits. The different alleles can arise from different methylation of DNA. To explain you in a better way, if we take two inbreds, the DNA methylation pattern is fast, but when the heterosis is concerned, when hybrids are produced, the methylation pattern is relatively so slow, because of which the activity of hybrids are considerably good. Therefore, it can be suggested that inbreeding depression partly or primarily result from the lower level or fewer genes expressed simply due to homozygosity of methylated DNA in regulatory factors, while heterosis is from higher level or larger number of genes expressed simply due to heterozygous condition between methylated and non-methylated DNA in the F1 hybrids. Next process is enhanced cellular energy in hybrids. Heterosis may be the result of greater cellular energy efficiency of hybrids due to selective protein synthesis and metabolism. About 1 to 5% of the genes of different species have alleles that encode unstable or inefficient proteins. It has been postulated that the quality control mechanisms in the hybrid detects the allele encoding unstable or inefficient proteins and suppresses their expression. This selective suppression of gene expression would lead to an overall saving of energy that would have otherwise been spent on the synthesis and degradation of these unstable or inefficient proteins. This energy saving would become substantial when it is summed up over all the loci. As a result, there would be more rapid cell division in the hybrids, leading to the growth advantage and ultimately improved performance. तो यहाँ पे इस स्लाइड में यही बताया गया कि जब हम हाइब्रिड बनाते हैं वहाँ पे सेलुलर एनर्जी भी जनरेट बहुत ही ज़्यादा क्वांटिटी में होता है बिकॉज ऑफ द प्रोटीन कॉम्प्लीमेंटेशन, मतलब एक प्रोटीन्स के बीच में फेवरेबल इंटरेक्शन बन जाता है। उसके साथ में हाइब्रिड्स में क्या होता है, रैपिड सेल डिवीज़न हो जाता है, तो वो बेटर परफॉर्मेंस और फास्टर ग्रोथ होने में डेवलप करता है और उसी के साथ में रिप्रोडक्टिव स्टेज भी जल्दी जल्दी आ जाएगा, सो दैट along with the greater amount or quantity of the outcome we may get the faster growth rate also. मीन्स यहाँ पे हाइब्रिड्स हैं हमेशा पॉजिटिव में भी और नेगेटिव में भी फैक्टर्स मूव करते रहते हैं। जैसे कि हमने अर्ली ड्यूरेशन सोचा, दैट्स ए नेगेटिव फैक्टर बट पॉजिटिव फॉर आर बेनिफिट, और अगर यील्ड की बात करेंगे तो वो ऑलरेडी हाइब्रिड विगर के टर्म्स में बात करते हैं। Coming to the increased ploidy level, aneuploidy reduces vigor while polyploidy are more vigorous than diploids. This you know when the polyploidy condition is exposed or when we increase the set of chromosomes in a genome. What will happen means when the ploidy level is increased, in terms what I want to say, that means there will be increased heterosis which is observed in some cases. That indicates that organisms have mechanisms to sense gene dosage balance. The dosage sensing is likely to be a genome-wide mechanism that is independent of specific alleles or loci, and this mechanism could contribute to heterosis. There are certain evidences for existence of variation in the different ploidy level. Some genes may be present in one line, but they may be absent from another line. Some genomic regions may be present in more than one copy in the genome of one line, while the genome of another line may have additional copies of some other genomic regions. That means heteroploids, which are having the different or high vigorous as comparing to the other ploidy conditions. In case of two inbred lines differ for the genomic regions showing presence or absence and showing copy number variation, their hybrid would have average copy number of all the genes involving in these variations. In comparison, the parental inbred lines would show the two extremes of copy numbers possible for them. Genome dosage balance in hybrids. One inbred line has 3 copies per genome of one genomic region, and the other inbred line has 4 copies per genome of another genomic region. This is just an example. These inbred lines will have 6 and 8 copies in one inbred of the concerned genomic regions, while their hybrids will have only 4 and 5 copies respectively of these two regions. So that what will happen? The hybrid would show greater gene dosage balance for the two genomic regions and would be more vigorous than the two parental inbred lines. The gene dosage effects over the entire genome will add up to generate heterosis. The findings of the study involving diploid and triploid inbred lines of maize indicate that the main parts of heterosis results from dosage sensitive mechanism. There is evidence that the mechanism of heterosis is significantly affected by dosage of that is observed in cotton genomes. Coming to the molecular markers' role in explaining the genetic basis of heterosis. In some studies, it is reported that the importance of overdominance through marker studies, especially in case of maize, the hybrid vigor is the evidence which is proved from over-dominance hypothesis, others have found dominance to be predominant, while some other observed to be extensive epistasis. Example, molecular markers linked to QTLs were used to analyze the expression of heterosis in elite rice hybrids, that is Zenshan 97 and Mingai 63. This you need to remember these two varietal name. One is Zenshan 97 and one is Mingyu 63. The QTLs showed overdominance, but the overall heterozygosity was not relevant for heterosis. Digenic epistasis was common, and even such pairs of loci that did not exhibit overdominance generally showed epistatic interaction. Thus, in rice, main contributor to heterosis were overdominance at individual loci and epistasis, while overall heterozygosity was not that important. This is proved by making the double hybrids also. The F1 hybrid performance was not always positively associated with heterozygosity, but it was explained by additive dominant, including partial complete overdominance, and epistasis, that is additive dominant, and dominant kind of gene interactions. That means heterosis cannot be simply or singly explained with the help of dominance theory. It's a combination of dominance, overdominance, even the epistasis in different crops. Coming to the importance of genetic distance in heterosis, genetic distance in short form is called as GD, that may be used to classify the lines or populations into distinct heterotic groups. Heterotic groups का ये मतलब होता है based on their vigor or their characteristics. जैसे कि tall planted है तो उसको एक heterotic group में identify या classify किया, या कोई different geographical region के basis पे उसको identify किया, या उसके yield के हिसाब से grouping किया. तो grouping कैसे किया जाता है? based on their dendrogram pattern. जैसे dendrogram में grouping आ जाते हैं कि उसका सब clusters कैसे characterize हो रहे हो, वो कैसे एक एक group में आ रहा है, based on that heterotic groups बनाते हैं. जैसे कि मैंने एक यहाँ पे dendrogram draw किया है, तो नीटली नहीं आ रहा है, लेकिन देखिए जो इतने लाइंस है या इनब्रेड्स है, ये सब एक हेटेरोटिक ग्रुप में आ गया. सिमिलरली अगर मैं दूसरा हेटेरोटिक ग्रुप बनाऊंगी तो ये सब दूसरे हेटेरोटिक ग्रुप में आ जाएगा. Heterotic group comprises such lines or population which when crossed with each other show little or no heterosis. But when lines or populations from different heterotic groups are crossed, there is moderate to high heterosis, which is often termed as heterotic pattern. तो heterotic pattern क्या होता है? The basic aim of making heterotic groups क्या होता है कि based on our needs, एक ग्रुप बनाएंगे तो उसका मतलब ही कुछ और होता है, तो दूसरे ग्रुप से वो कंपेयर कर सकते हैं, मतलब कैरेक्टर्स बिल्कुल डिफरेंट होते हैं. तो ऐसे ग्रुप्स बनाने का क्या मतलब है? हमारे कैरेक्टर जिसमें हम इंटरेस्टेड है उन कैरेक्टर्स को लेके उसको हाइब्रिड में इनकॉर्पोरेट करने में ये बहुत ही हेल्पफुल होता है. In case of maize, heterotic groups are well established. Typically, the inbred lines of heterotic groups have high constitution, high characterization. Coming to the overall mechanisms that have been proposed to explain heterosis, मतलब मैं molecular basis of heterosis को summarize कर रही हूँ with the help of different mechanisms. पहला है intermediate amount of biochemical product at optimum level. इसको मैंने एक्सप्लेन किया था विद द हेल्प ऑफ पी अमाइनो बेंजोइक एसिड कंटेंट इन न्यूरोस्पोरा क्रसा. तो क्या हुआ था, जब ये कंटेंट कम था तो ग्रोथ ठीक से नहीं हो पा रहा था, तो एक इंटरमीडिएट अमाउंट, मतलब ज़्यादा भी नहीं होना चाहिए कम भी नहीं होना चाहिए. जब हाइब्रिड बनाया तो इंटरमीडिएट अमाउंट ऑफ पी अमाइनो बेंजोइक एसिड प्रोड्यूस हो गया. बिकॉज ऑफ दैट न्यूरोस्पोरा क्रसा हैड फास्टर ग्रोथ रेट. सेकेंड मैकेनिज्म है द प्रोटीन प्रोडक्ट्स ऑफ डिफरेंट अलील्स विथ डिफरेंट प्रॉपर्टीज और फंक्शंस. मतलब एक इनब्रेड में एक काइंड ऑफ प्रोटीन है और दूसरे में दूसरा है, लेकिन प्रेजेंस ऑफ उन दोनों प्रोटीन्स के प्रेजेंस के हिसाब से बहुत ही अच्छा फंक्शन हो सकता है. एग्जाम्पल सिकल सेल हीमोग्लोबिन इन ह्यूमन, तो होमोजाइगस कंडीशन में होगा तो सिकल सेल अनीमिया कॉज करेगा. अगर वो हेटेरोजाइगस कंडीशन में होगा तो मलेरिया रेजिस्टेंस कॉज करेगा, ठीक है। थर्ड एग्जाम्पल है जनरेशन ऑफ हाइब्रिड प्रोडक्ट। एग्जाम्पल मैंने कोट किया था, जैसे कि ट्रांसक्रिप्शन फैक्टर एक्ट करता है। जब हाइब्रिडाइजेशन है तो ट्रांसक्रिप्शन फैक्टर एक्टिवेट हो जाएगा, वो प्रमोटर से बाइंड कर जाएगा एंड इट विल एनेबल द ट्रांसक्रिप्शन फैक्टर। जैसे ट्रांसक्रिप्शन फैक्टर एक्टिवेट हो जाते हैं और ट्रांसक्रिप्शन हो जाता है तो आप जानते हैं ट्रांसक्रिप्शन से डेफिनेटली आरएनए बनता है, तो प्रोटीन प्रोडक्शन आसानी से बन पाता है, तो मेटाबॉलिक रेगुलेशन पाथवे एक्टिवेट हो जाता है। फोर्थ मैकेनिज्म है इफेक्ट्स प्रोड्यूस्ड इन टू डिफरेंट टिश्यूज़ को एक्सप्लेन किया था, ADHS एंड ADHF जो मेज डिफरेंट टिश्यूज़ में हुआ था, तो वो दोनों के एक्सप्रेशन से बेटर कैरेक्टर्स पाए गए थे। फिर नेक्स्ट है एनहांस्ड एक्सप्रेशन लेवल्स ऑफ सम जीन्स, तो ये रिलेट होता है हिस्टोन मॉडिफिकेशन से या हिस्टोन रेगुलेशन से और आरएनए इंटरफेरेंस के एक्टिविटी से। फिर एक्सप्रेशन ऑफ ग्रेटर नंबर ऑफ जीन्स, ये भी रिलेटेड है हिस्टोन मॉडिफिकेशन और एपिजेनेटिक चेंजेस से। देन सुपीरियर जीन डोसेज बैलेंस, जैसे कि नंबर ऑफ जीन का डोसेज बढ़ेगा वैसे एक्सप्रेशन पैटर्न भी बहुत ही कंट्रोल में आ जाएगा। देन इंक्रीज्ड एनर्जी एफिशिएंसी, हाइब्रिड्स में ड्यू टू बेटर मेटाबॉलिक बैलेंस एनर्जी एफिशिएंसी भी बढ़ जाता है। देन इंक्रीज्ड कनेक्टिविटी इन द मेटाबॉलिक नेटवर्क्स, मतलब मेटाबोलाइट्स का जो प्रोडक्शन होता है वो एक बैलेंस्ड वे में चलने लगता है। तो ये सब जो भी मैंने प्रोसेस एक्सप्लेन किया है उसको सब समराइज़ करने के लिए ये टेबल दिया गया है। दिस इज़ मॉलिक्यूलर चेंजेस एट एपिजेनेटिक, जीनोमिक, प्रोटियोमिक एंड मेटाबॉलिक लेवल विच लीड टू हेटेरोसिस ट्रेट्स। तो मॉलिक्यूलर चेंजेस किस किस में आता है, स्मॉल आरएनएस में मिथाइलेशन एंड क्रोमैटिन मॉड्यूल रेगुलेशन से। तो ये ओमिक स्टडीज़ है, तो इसमें और भी आता है ट्रांसक्रिप्टोन, सिस एंड ट्रांस रेगुलेशन, फिर प्रोटियोमिक्स एंड मेटाबॉलिक्स आता है। फिर ये नेटवर्क किसके ऊपर काम करता है, एपिजेनेटिक जीन एक्सप्रेशन एंड नेटवर्क चेंजेस के ऊपर। तो ट्रेट एक्सप्रेशन में ये दिखाता है शुगर, स्टार्च एंड मेटाबोलाइट्स एंड बायोमास एंड यील्ड। तो इनके बेटर कंपैटिबिलिटी से हेटेरोसिस इन द डिज़ायरेबल ट्रेट्स हो सकता है। I hope this information is helpful for you. If you like this video, please do not forget to hit the like button. Please subscribe my channel. Keep on liking, commenting, sharing. Thank you once again.